This recording is part of a series recording covering the heart, and it's going to address action potentials in cardiac muscle. So previous recordings, we've covered the cardiac conduction system. So here represents the SA node, which is the pacemaker, under normal instances, the pacemaker of the heart. So we talked about the slow response action potentials that we saw in the SA node, and I'm going to show it to you again just to remind you. And then that impulse, the impulse originates in the SA node, but then it's carried over to the atrial muscle, travels down here to the AV node, and then the AV node, you got the bundle of His that goes through the annulus fibrosus, which is here, and then the impulse travels down your bundle branches, and then you've got your terminal Purkinje fibers. Now that's going to allow that electrical impulse to make its way to ventricular muscle. So the muscle needs to be able to generate an action potential in order for it to contract. So we're going to be looking at how the action potentials look specifically within the ventricular muscle, which is an example we call a fast response action potential. Now here, this is what a slow response action potential looks like. This is what you saw within the SA node. So key things to remember: the membrane resting potential is less negative in the SA node because those cells are leakier to sodium. Plus, you don't have a true rest. It never really stays down here. It just slowly creeps up, again, because of this slow influx of sodium, because those cells are leakier to sodium. Once enough sodium has entered and you reach threshold, you will generate an action potential, but it's not as fast as what you would see, say, within ventricular muscle or even atrial muscle. But that upstroke that we see, that depolarization that you see here during the action potential, is actually due to the movement of calcium inside the cell. The reason for that is because the sodium channels are already inactive at this membrane potential, so it's actually the movement of calcium into the cell. You have that repolarization. This is not going to change every time we've ever talked about repolarization. It's because of potassium leaving the cell. So this kind of slow, kind of like sodium coming in, and then we just generate an action potential, that's going to set things off. It's going to set off the, start opening up voltage channels in atrial muscle, and there'll be voltage gated sodium channels, and it'll travel down parts of the conduction system and eventually set off the ventricular muscle. So we're going to look at what's involved in how that ventricular muscle action potential looks. So this looks different from what you've seen before. It looks different from that SA node. It looks different from when we looked at nerves. It looks different from what you saw in skeletal muscle. So this is a fast response action potential that we see in ventricular muscle. So let's go through some of the steps. What's involved? What do you see with these phases? So I'm actually going to start here with phase 0. So phase 0 is the upstroke. What you see is rapid depolarization. Look how quick. It's like straight line up. This is due to the opening of sodium channels. And when those sodium channels open up, you're going to have this massive influx of sodium inside the cell, and you'll notice you see a reversal of charge. So you go from minus 90 millivolts to a positive value. Those sodium channels are actually, that opened, are actually referred to as fast sodium channels. And so if I want to draw a picture up at the top that represents movement of ions from ECF to ICF or vice versa in phase 0, I'll use the gray, since it's kind of gray, is I'm going to go like this. So sodium is going inside the cell because, again, there's a lot more sodium out than in. You open up a voltage-gated sodium channel, you have a massive influx of sodium inside the cell. Then what you see here at 1, it's a slightly darker gray. I'm going to change it to green so you can really see it. So phase 1 is the notch. And the notch is actually due to an initial repolarization of the cell. We have, it says here, even has it in here, transient potassium channels open, and potassium starts to leave the cell because you have a lot more potassium inside the cell. And so it starts to go back down to that 0 millivolts. Phase 2 is the plateau phase, and I'm going to change the color again. Here's phase 2. But I got to go back to phase 1 and draw it on the top. So with phase 1, what I'm going to draw is I'm going to draw some potassium. Leaving. Okay, so I'm going to say a little bit of some potassium started to leave. Now in phase 2, what I'm going to do in gold, that is the plateau phase. Now, having that plateau phase, you notice that membrane potential is not changing. It's staying the same. Why is it? Because I have an equal number of positive charges coming into the cell as I do leaving the cell. And so what's coming in, what's going out? So what's coming in is calcium. And calcium is going through what we call L-type calcium channels. They're long-lasting type of calcium channels. And those are important channels. Okay, I'm going to mention to you what them, you've actually, believe it or not, you know what they look like, or you've probably heard of them, just don't realize it. So we have calcium channels, these L-type calcium channels open. Calcium starts to move into the cell. And I want to remind you, cardiac muscle does not have well-developed sarcoplasmic reticulum like you do in the skeletal muscle. So calcium that is needed to contract in cardiac muscle, some of that calcium is coming from outside the cell. And so that calcium coming in is important to be able to have that for muscle contraction. You have calcium from the sarcoplasmic reticulum, but you also have calcium coming from the extracellular fluid. So you have these positive charges coming in, but at the same time is we've got some potassium leaving. So you have potassium efflux. And they're actually through these channels. You don't have to write this necessarily down, but they're called delayed potassium rectifier channels. So they're a little delayed. So we have calcium coming in, potassium moving out. The total number of charges leaving versus coming in is the same, and so we have a plateau phase. Now the calcium channels remain open for a long period of time, a little longer than what most people think. That's why you have this little plateau here. And then during phase 3, which we're going to keep as red, what's happening in phase 3 is we have potassium is going to leave the cell. So phase 3, what's happening is the potassium channels are opening up, or sorry, I should say, potassium channels open up, and the calcium channels have closed, or closing. So calcium channels close. Now we have these potassium channels that are opened up, and potassium is leaving the cell. And so we're getting back to that membrane potential. And so at 3, we can say what's going on. This is repolarization during phase 3. So phase 0 is the upstroke, that we have rapid depolarization. Phase 1 is the notch. We've got an initial depolarization, initial repolarization. 2 is the plateau phase. Phase 3, we're repolarizing. Phase 4 is resting membrane potential. So resting membrane potential, you have that stable membrane potential What's allowing that to be stable is you have the sodium-potassium pump working, it's going to get the sodium to come back out. We're going to pump 3 sodiums out of the cell in exchange for 2 potassiums. We're going to keep that unequal distribution. It's that membrane potential is negative. It's closer to the potassium because remember, all membranes are a little bit more permeable to potassium. So we're going to be a little bit closer to the equilibrium potential for potassium. So at rest, got a stable membrane potential about -90. Something comes along to stimulate those voltage-gated sodium channels. And here we call them fast sodium channels. They open, and then you see the beginning of the action potential right there. Now here, this I want to show you is in cardiac muscle, and this is very similar to what you saw in skeletal muscle. You should have learned about some T-tubules, the T-tubules, and you learned about the terminal cisterna of the sarcoplasmic reticulum. Sarcoplasmic reticulum has all this calcium in there. You needed that calcium to go flood into the sarcoplasm, so we can use that to bind to the troponin C. That troponin C, once it binds to calcium, it moves tropomyosin out of the way, and so the binding sites on actin are now exposed so myosin can interact with it. Well, similar thing has to happen in cardiac muscle, but there's a slight difference here. Is you have an action potential that's generated, and this calcium here coming from the extracellular fluid is going to go through these channels right here, and those are the L-type channels. And you may have heard of them referred to as DHP receptors. So in in cardiac muscle, the DHP receptors are these L-type calcium channels, and they're actually just calcium channels. Calcium moves through those, and it creates allows the ryanodine receptors that are in the sarcoplasmic reticulum, which are calcium channels too, allows them to open, and the calcium from the sarcoplasmic reticulum is going to flood out. So in this case, in cardiac muscle, the calcium coming in from the extracellular fluid through the DHP receptors is going to trigger the opening of ryanodine receptors, which are calcium channels. And we're going to have this whole bunch of calcium inside the cell. I need calcium from the sarcoplasmic reticulum as well as the extracellular fluid so I can use it for muscle contraction. Now in skeletal muscle, if you learned about the DHP receptors, you learned that they were responsive to changes in membrane voltage, and they usually are blocking these ryanodine receptors, but that voltage coming down here causes them, the foot process of them, to pop open, and so those the ryanodine receptors open up. Now, once we've done what we need to do, that calcium, some of that calcium is going to be pumped back into the sarcoplasmic reticulum using a calcium ATPase, and we also have some of the calcium that's going to leave the cell through a sodium-calcium exchanger. The sodium-calcium exchanger works because that sodium-potassium pump also creates a favorable gradient for sodium to come in. So this here is actually an example of a secondary active transporter. So when all's done, we're going to put the calcium back in the sarcoplasmic reticulum, pump some of it out. We're going to wait for another time so we then get that calcium to come back in the cell, and we can use it for muscle contraction. Now here, I do want to mention refractory periods. So I'm showing you a comparison between cardiac muscle and skeletal muscle. Cardiac muscle has a much longer absolute refractory period than you do with skeletal muscle. The reason for that, there's a reason why we want that, is ventricular muscle having that longer refractory period is going to allow muscle contraction to take a little bit longer. But also having that long absolute refractory period is going to prevent tetany from taking place. No matter how frequently the stimulation is, the cardiac muscle should not reach tetany like we do in skeletal muscle, because if it's in tetany, then you're not going to be able, the heart won't be able to relax, be able to fill up with blood. So the refractory periods in cardiac muscle are much longer than you see in skeletal muscle. So remember, absolute refractory period. During that period of time, no matter what the force of stimulation is, another action potential will not occur. The yellow represents a relative refractory period. So you could generate another action potential, but it has to be a much higher than normal stimulus for it to take place, because during that period of time, some of the voltage-gated sodium channels have now reset, and then we could generate another action potential during that period of time. Now, there actually is a difference in refractory periods within atrial muscle versus ventricular muscle. And a important thing to, a little tidbit of information, is because atrial muscle has a shorter refractory period than ventricular muscle, it has the ability to contract at a faster rate than ventricular muscle. Under normal conditions, they're contracting at the same rate, because that's important. But in some instances, when people have some arrhythmias, the atrial muscle is actually contracting faster than the actual ventricular muscle. So we're going to do one last thing, and then we're done. So for all this stuff to take place, it requires energy. You need ATPase for the sodium potassium pump. You need ATPase, or ATP for the calcium pump. You need ATP for muscle contraction. So you need energy. Cardiac muscle must use aerobic metabolism. It needs oxygen. Does not like to work anaerobically. And so it's been adapted to be very aerobic. A lot of mitochondria, people say the powerhouse of the cell, that utilizes oxygen to be able to generate a lot of ATP. Myoglobin is a pigment that binds to oxygen. So in case we're not getting quite enough blood flow to that cardiac muscle, we got a little backup. So we got it's an oxygen binding protein there in cardiac muscle. Fairly good capillarity in cardiac muscle. And the cardiac muscle will pretty much use whatever you give it. It's like, you got me some fat, I'll use some fat. Got me some glucose, I'll use some glucose. Got some ketone bodies around, I'll use ketone bodies. So it's pretty good at using what's available. It'll even use lactic acid. It's like, what? Someone say, well, I learned lactic acid is a product of anaerobic metabolism. Yes, it is. Your skeletal muscles could technically be working anaerobically, producing lactic acid. Your red blood cells are always producing lactic acid because they can't work aerobically. They don't have any mitochondria. So as long as the heart has sufficient levels of oxygen, it can actually take the lactic acid, convert it back to pyruvate, and then use that using aerobic means. It converts it to something called the acetyl coenzyme A that goes through what we call the TCA cycle. Then go to electron transport chain, and you'll be able to get some energy from it. So long as it's working aerobically, it can take lactic acid from another source and actually use that for energy too. So it's going to use whatever you give it, because it's like it needs quite a bit of energy. So it's adapted for that. So what would potentially happen if I don't have good blood flow to the heart? It really, really, really loves to work aerobically. It's not going to like you. Things aren't going to work very well if it can't get the oxygen that it needs. So that could lead to hypoxia, or I should say ischemia can lead to hypoxia, which could lead to a heart attack. So I'm going to leave it there, and next recording, we're going to look at the electrocardiogram.